Okay, November 19, paper 6, variant 2. Let's read together. A student investigated the effect of enzyme amylase on the breakdown of starch suspension. The starch suspension was placed inside a bag made from dialysis tubing. And then he gave you the equation. Starch using the amylase enzyme changes into reducing sugar. Okay. So if I am investigating the effect of enzyme amylase, I might put amylase in one test tube and the other test tube without amylase, for example. Okay. The dialysis tubing is made from a type of membrane that is partially permeable. Dialysis tubing is partially permeable. What does partially permeable means? He told me, only small molecules can pass through this membrane while the large molecules cannot pass through. Okay. 2 large test tubes were put in a water bath. Each large test tube contained 20 cubic centimeters of distilled water, and the temperature of the water bath was 40 degrees. Since you put the same volume of water in the 2 test tubes and you put both test tubes in the same temperature, I can say that these are controlled variable. This is a controlled variable. This is also a controlled variable. Okay, what about the water bath? What do we have to say about the water bath? Is this accurate? So the water bath here is a source of error. It's a source of error because the temperature is not maintained. If you leave it on the table, this 40 degrees will decrease. So I can put a line كده and I write source of error. ماشي. A knot was tied at the end of one piece of the dialysis tubing to form a bag. Here, the dialysis tubing, he tied the knot at one end. 5 cubic centimeter of starch suspension was put into the dialysis tubing bag. Inside the bag, he put 5 cubic centimeter of starch suspension. The contents of the tubing were mixed well, and outside the bag was rinsed with distilled water. He washed the bag from the outside with distilled water. Okay. He repeated the steps using 5 cubic centimeter of distilled water in step 4. Okay. Instead of the amylase solution. Sorry, we skipped step 4. هنا في step 4 قال لي إن he put also amylase. يبقى in this test tube he put starch and amylase, while in this test tube he put starch, same volume of starch. So this is also a controlled variable since he put the same volume of starch in both, while instead of the amylase he put distilled water. Okay. The dialysis tubings were placed in large test tubes as shown. This one contains amylase and starch. This one contains water and starch. Both test tubes were placed in a water bath, left for 10 minutes. After 10 minutes, they were removed. The contents of the 2 test tubes and the dialysis tubing were tested for sugar and starch. What will I do here? Inside this bag, there is starch and amylase. Inside this bag, there is starch and water. What will happen? In the first test tube, there is enzyme with the starch. Firstly, if you did the iodine test inside the whisking bag, you will find starch because you already put starch inside. If I did the iodine test to the water outside, am I going to find starch? No. Why? Because the starch is a big molecule. It cannot diffuse out. What else? I also put amylase. The amylase will break down starch into small sugars. So if you did the Benedict test inside for the contents inside the whisking tube, you will find sugar present. Also, sugar can diffuse out of the membrane. So if you did the Benedict test outside, you will also find sugar outside. What about this test tube? If I did iodine test inside, you will find starch. If I did iodine test outside, you will not find starch. Why is this? Because the starch is a large molecule. It cannot diffuse out of the membrane. Type, am I going to find sugar inside or outside in this test tube? Am I going to find any sugar in this test tube? What do you think? يبقى no, in this test tube, there is no sugar. Why is this? Because there is no enzyme to break down the starch into sugar. I put water instead of the enzyme. Okay, these state the name of the solution that the student would use to test for starch. What is the name of the solution to test for starch? And starch, not sugar, starch.
Iodine.
Iodine.
Iodine. Okay. Describe how the student would test the substances for reducing sugar بقى. For reducing sugar, what do I do to test for reducing sugar?
Benedict.
Benedict test, add Benedict solution. And then what's the second step after you add Benedict? What must I do after?
Heat.
Heat.
And,
Heat in water bath. Of course, if the result is positive, it will change to red. Suggest why the outside of the dialysis tubing was rinsed in step 5. Why did we rinse? Why did we rinse the outside of the whisking tubing with distilled water? What will I say? To remove the starch from the outside. After you put the starch, ممكن يكون some of the starch may spill on the outside of the whisking tubing. So you have to remove it in order not to give you false results. Okay? يبقى to remove any starch on the outside. Okay. This is the results there. These are the results. He gave you the observation. Dialysis tubing 1, which means he tested the contents inside. The starch test was brown, okay? No starch. While the reducing sugar test was brick red. Reducing sugar test inside was red. طيب. Why is the starch test brown? I said at the beginning that I put the starch with my hands inside the whisking tubing. Why is the starch test brown now here? In the first dialysis tubing, you put enzyme with the starch. The amylase broke down the starch. So if you did the iodine test, it will be negative. There's no starch. What about the sugar? It is brick red, which means positive. There is sugar inside the tubing. Where did it come from? We said that the amylase breaks down the starch into sugar. So here there is sugar. Type. What if you took a sample from the outside, from the water outside, and you did a starch test? Of course, it will be brown, okay? As we said, the starch is a large molecule. It cannot diffuse out. Also, the starch was all broken down by the amylase enzyme. But the sugar, if you did a sugar test or Benedict test outside, you will find it orange, which is still positive. Why is this? The sugar is a small molecule, so it can diffuse out of the test tube. Okay, what about the results for the second one? For the second one, you put water and starch inside. If I did an iodine test inside the whisking tubing, I will find it positive. It's blue-black. If I did a reducing sugar test inside, it is blue. Why is it blue? In this test tube, the reducing sugar is blue because You did not put enzyme, so the starch was not broken down. Outside, in the water outside, the starch test is brown. Starch cannot diffuse out because it is a large molecule. Reducing sugar is also blue. There was no sugar from the beginning to diffuse because there is no enzyme. Okay, the first question. Prepare a table and record the results of the starch and the reducing sugar test in the table. The first thing to think about, what did I change in this experiment? To record your results in a table, we always put the changed variable on the left side. What is the changed variable in this experiment? One you put amylase and the other you put water. So this is what I did. Enzyme presence or absence. Here I put amylase and here I put water. On the right side, it is always the results. Right side, results. What are the results you were observing? The tests. Starch test and reducing sugar test. But take care. For the starch test, you did it here inside and outside. And the sugar test, you did it inside and outside. Also here, inside, outside, inside, outside. So this is why after the amylase, I put the site of the test. Is it in the dialysis tubing or the test tube? Here, same thing, dialysis tubing, test tube. And then you fill the results back. Starch test in dialysis tubing 1 was brown. Reducing sugar test was brick red. يبقى dialysis tubing 1, الstarch test was brown. Reducing sugar test was brick red. For test tube 1, it was brown and orange. Brown, orange. For the water, for the test tube بقى 2 بقى هنا. Blue, black, and blue. Blue, black, and blue. Test tube 2, it was brown and blue. Brown and blue. Okay. Now the table is finished. The question after is, state 3 conclusions for the results. The 3 conclusions in this test, I can choose anything. Number 1, I can tell him that in test tube 1, there is no starch. There is no starch as it broke down by amylase. Also, I can say that there is a sugar in test tube 1. There is sugar produced. What about test tube 2? In test tube 2, the sugar produced inside and it diffuses also. I can add that after the sugar was produced from the amylase that broke down starch, it diffuses out as it is a small molecule. Sugar is produced, which diffused out as it is a small molecule. The last thing, in test tube 2, there is negative starch test. In test tube 2, no starch. Why is this? Because as we said, as starch is a large molecule that cannot diffuse. It's a large molecule, it cannot go out. Okay. State 2 variables that were kept constant. What are the two variables that were kept constant? We already marked a few things.
Volume of water
The volume
temperature.
of water, okay, they put 20 cubic centimeter, the temperature, the volume of starch, all these are controlled. Okay, so volume of water
Starch.
and temperature. Also, I can say the volume of starch suspension. Okay. Plan an investigation using the dialysis tubing to determine the effect of temperature on the activity of the enzyme amylase. Again, in this investigation, you want to test the effect of temperature on the activity of the amylase. So you will do something like the experiment he did, but you will change a few things. Number one, you always tell him how many test tubes you will bring, for example. So we said use
5 test tubes.
5 test tubes
Using the slim
and
tube
put 5 dialysis tubing in them, like what he did in the previous experiment. He got the test tubes and he put the dialysis tubing in them. Okay. Then you tell him what are the controlled variables in your experiment. Since you are going to change the temperature, everything else should be controlled. Put same volume of amylase, same volume of starch suspension. What else can I say? I can put same concentration of amylase, and yes, same concentration of amylase and starch, same pH, anything that affects the enzyme should be the same. Same volume of water in the test tube as he did, same volume of distilled water in the test tubes. All these factors should be the same. Okay.
بس دي بس سؤال.
Yes.
بس هو احنا
Third point is which factor will you change? You will say put each in a water bath with different temperatures. And you give him an example. For example, 10, 20, 30, 40, 50 degrees Celsius. Okay? And then you tell him what are the results you are going to measure. What results am I going to measure here? I will do iodine and Benedict test like he did, but I can use one. For example, you will tell him that every minute you will take a sample from inside the whisking tubing and do an iodine test. Why am I doing this? To make sure that the starch is still there or is it finished? Of course, with the optimum temperature, starch will be finished quickly. So after a few minutes, you will find the starch test negative. While at low temperatures and very high temperature, the enzyme denatures. So if you keep doing the iodine test, it will always be positive. Starch is always there, it is not digested. So how will I write this? Take a sample from inside the whisking tubing every minute and do iodine test to test for presence of starch. Okay? Record the time taken for all starch to be digested. This is how you will know which temperature is the best. Okay, number 5, you can tell him an improvement for the experiment. Since you are putting each in a water bath, what is the improvement for this step? What should I do with the temperatures?
مش فاهمة ايه سؤال
Use
حضرتك.
thermostatically
Yes,
control.
you cannot leave the water bath as they are like this on the table because their temperature will change. So you have to tell him, maintain the temperatures of water bath using thermostatically controlled water bath. Number 6, you put a safety precaution like wear goggles. Always the goggles are important in any experiment. And then number 7, you tell him repeat experiment at least 2 more times and take an average. G. The student wanted to find out if the amylase enzyme passed through the dialysis tubing into the large test tube. Amylase is made up of a protein. How would the student find out if the solution in a large test tube contained protein? What do I do to check if there is protein? I will tell him, do the Biret test, add Biret reagent. What is the positive result? If there is protein, what will happen?
To be purple.
Yes, if there is protein, it will change to purple. Okay. This is it for question 1. Let's see about question 2. An investigation was performed to determine the effect of light intensity on the leaf size. Investigate or determine the effect of light intensity on the leaf size. You want to see if you change the light intensity, what will happen to the leaf size. So this is the factor changed. The light intensity is the factor changed. Okay. Plants were grown in 3 different light intensities. The maximum width was recorded and the average value was calculated. Okay. The results for the 3 leaves grown in high light, there is a horizontal line, indicates the maximum width. What are you going to do? He gave you the leaves for low light intensity, medium light, and high light. But it is missing 3 leaves, and he wants you to calculate the average. Where will I get these 3 leaves? You will just measure with your ruler these 3 lines. Of course, take care of the unit. When you measure with your ruler, you are measuring in centimeter, but here he wants it in millimeters. So you have to multiply by 10. You will put the 3 numbers here, and then you will calculate an average. So the 3 numbers here will be around, after you measure them, 26, 31, and 26. How do I calculate the average? I add all these numbers together, and then I divide by how many numbers did I add? How many numbers? 8 numbers. So I will add all these, and then you'll divide by 8. You will have the answer, which is 28. Okay, calculate the average width. We already did. Plot a bar chart of the average leaf width in low, medium, and high. He wants a bar chart of the average width. So here, the first thing you will do is put the labels. What are the labels? You have 3 bars, light intensity, low, medium, and high. What are you measuring is always on the y-axis. I will put the average leaf width. This is the results I'm measuring. And then you will check the numbers. The largest number is 41. So when you put the scale, you have to cover after 41. Here I did 0, 10, 20, 30, 40, and 50. And then you start drawing a bar for each light intensity. Here it was 14, 41, and 28. But take care, bars have to be equal and the spaces between them are equal. Okay? Circle one measurement that could be considered anomalous and give a reason for your choice. I want you to check all the numbers here and tell me which one is anomalous, which might be a wrong result. The wrong result here will be 12 in the medium light intensity. Why is this? Because if you check كده, all the width of the leaves was 40s, 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 40s. This was the only one that was 12. So definitely there is something wrong that happened here. Why did I circle this? I will tell him. Erase the other circles back. You only circle the wrong answer. I will tell him that it does not fit the pattern of result. It does not fit pattern of results. I can also say that there is a big difference between it and between the other numbers. Okay, what is the variable that was changed in this investigation? What did I change? I already underlined it. What did I change? What was I testing in this investigation?
The light intensity.
The light intensity. طب what was the factor measured? What was the results I'm measuring?
And the leaf size.
The maximum leaf width. Okay, so what was I measuring? First, what was I changing? Light intensity. While the variable that I measured was the maximum leaf width. Okay. He then gave me a cross section of a root and he asked me to draw a large drawing to show the different areas. Do not draw individual cells. Okay, what do I need to draw here? Here I have
And
different
then
layers.
4 layers.
Yes, there is a thin layer. Let me draw it in yellow so you can see. Thin layer, this one. And then there's a lighter layer, this one. And there's a dark layer and a circle in the middle with a star shape in the middle. These are the layers that I have. So what's important here, one second. What's important is that the drawing should be big. You have to draw the layers in proportion. How? Like this. The first one is thin, and then a thicker one, and then the thickest layer, and then there's a circle, and then there's a star in the middle. Okay. Of course, after the drawing, he will ask you a magnification question. Measure the line AB in millimeters. Where's line AB? Here, you will measure it with your ruler in millimeters, and then you will put the number here. The actual diameter is 2 millimeters. Put the triangle. Magnification equals image over actual. Calculate the magnification. So you will get the number you measured over 2 to obtain the magnification. Okay. Last question. This is a cross section of a stem. He asked you, give me 2 differences between the root and the stem. You will look at this picture and you will look at this picture and tell me. The differences between the 2 pictures is that the root is more rounded in shape, more rounded. Here it is more circular, while this is a bit oval in shape. Number 2, the xylem vessels are in the middle, in root. Xylem are central. They are in the center here. They are in the center, while in the stem, the xylem vessels are on the periphery. I can also tell him that the xylem vessel in the root are larger, their size. They have a larger cross section. Okay. Last question, Babgad. A student suggested that measuring the leaf area is better than measuring the width. How the area of the leaf could be measured? So in order to count the leaf area, draw outline of leaf on grid, like a graph paper that is divided into squares. You draw the outline on the graph, okay, or on the grid, and then count squares covered by leaf What if half of the square is covered? You only include the squares that are more than half covered. Include squares that are more than half covered. Okay. Why is measuring the area better than the width?